What would you say is the world's deadliest animal? Sharks? Snakes? Humans? When it comes to killing humans, no other animal even comes close to mosquitoes. Mosquitoes carry devastating diseases. The worst is malaria. For perspective, nearly one human life is lost to malaria every minute. So, what is malaria? And why is it so lethal? Malaria is a life-threatening disease caused by Plasmodium parasites that are transmitted to people through the bites of infected female Anopheles mosquitoes. The alarming statistics is that more than 90% of the total malaria deaths are disproportionately contributed by the WHO African region. And almost 100% of these death cases in Africa is caused by this specific Plasmodium species called P. falciparum. You see, malaria is a completely curable disease, even though there is no effective vaccine against it yet. Hence, what is really driving the high death rates in Africa, and by extension the world, is the poor and costly diagnostics. Firstly, high-end equipment like flow cytometer are too expensive and bulky. Secondly, manual counting by a trained microscopist is required. Furthermore, considering the workload in Africa and the time required for each microscopic examination, this amounts to a really huge economic effort. Thirdly, microscopic diagnostics is not standardized due to differences in the experience and skill of the microscopist. Currently, this workflow is the gold standard for clinical malaria diagnosis. The microscopist has to manually count each malaria parasite, which is very labor-intensive and time-consuming. Herein, we propose to implement a deep learning model called MASK-RCNN, which is able to automatically detect and quantify different stages of P. falciparum infections more quickly and accurately. Given an input image, the prediction primarily involves 3 stages. In the first stage, region proposals are generated. In the second stage, classifier hits are being run on the region proposals to generate class probabilities and bounding box regressions. In the third stage, the mask hit is run on the detections to generate segmentation mask for every instance. How do we use this platform? Simply upload as many images as you like, and with just a simple click, the results are auto-generated for you. You can also generate selective masks for better visualization. From the results, we are able to achieve a high accuracy of 85%. Our model also uses significantly less time on average to analyze each image. It only takes 2 seconds as compared to 1 minute by manual counting. That is a lot of hours saved in the long run. Consistency in diagnosis will be higher too due to greater standardization in classification. As such, trained microscopists are no longer needed. Required skill level is therefore low. Consequently, we are able to operate at a lower cost as we save on time and manpower. And most importantly, our model shows much potential for scalability. Unlike old algorithms, we have demonstrated that we are able to detect subtle and less common differences between stages. We can also expand our work to identify leukocytes, other parasite species, and even other diseases which will definitely be useful for a medical diagnosis, especially in developing world settings. Our lab has already constructed a cheaper and portable image-based cytometer. For full deployability in the field, we intend to make further improvements by automating the microscopy process and complementing it with our MASK-RCNN model. Let's not forget that malaria is the deadliest disease. Diagnostic performances must improve at a faster pace than malaria is claiming lives.